So, welcome to another community meeting. I will start by actually presenting our conference. I have a slide here. Give me a second. Perfect. So, very few details. You probably, all of you joining here, you're aware, but also we're doing it because it's going to be a recording and it's going to be uploaded on YouTube. So we have updated our banner. The first conference of SVerse is happening this September. Registrations are open, and we also accept abstracts for talks, posters, workshops, tutorials, everything. And we have some slots left, but it's kind of first come, first served. I mean, some things will be evaluated, but the faster, the better. It's going to be September 10 to 12, and we have already confirmed 4 of our invited speakers. Probably you're aware of all of them. And yeah. And we have, so the deadline for early registrations is 15th of August. So please join us. We're happy to meet you all there. Core team is going to be there. We have a lot of things to discuss and to talk about. And the workshops will have tracks. So you can also, if you submit for a workshop or tutorial, you will probably kind of attract the relevant audience. Industry will be there. We have already people funding us and sponsoring us, and a lot of industrial partners joining us. So it will be from academia and industry as well. Registration's open. Go now. So that's from the SVerse side of the conference. And very quickly, I will pass to our speaker. So we have Quentin today, who is a PhD student at, so forgive my friends, right? At Mike's laboratory, Paris-Saclay University, and Gustave Roussy Institute. He will repeat after me, so we make sure that's the right pronunciation. He's mainly developing deep learning tools for multi-omics data, for precision medicine in oncology, and he has a special focus on spatial transcriptomics and single-cell resolution data. Today, he will be talking about Scyan, which is a method for fast cell population annotation, just with prior biological knowledge without require any training, label training. Quentin, if you share your slides, I can confirm that we can see everything, and the floor is yours.
Thanks for the introduction. Can you see it?
Yes, everything's great.
Okay. So thanks everyone for being here. Today I'm going to talk, as Michaela said, about Scyan, which is a biology-driven deep generative model for cell type annotation in cytometry. First, I'm going to quickly explain what is cytometry. So cytometry is a measure of proteins or markers expressions at the single cell level. So basically you have a table of size, number of cells times number of proteins. It's in terms of data structure, we can use ANData. And we have many types of cytometry techniques. For example, flow cytometry, which can measure up to 10 variables per cell. Spectral flow, up to 40 variables. Mass cytometry or cytof, which can measure up to 50 variables. So it's the largest amount of proteins that we can measure. And the last one is imaging mass cytometry, which is very similar to cytof, but we have some spatial information. So we can recover the coordinates of the cells based on the image. Now I'm going to quickly explain why I worked on Scyan. So the very traditional approach to annotate cell types in cytometry is to do what we call manual gating. The idea is that we will do many 2D scatter plots, like this one on the right, and we will select some cells on this scatter plot. For example, here we plot CD8 over CD4, and we annotate these cells, like the CD8 positive cells and CD4 negative cells, as being CD8 T cells. So this is really the traditional approach. It's very easy to understand how it works. It's very visual and intuitive, but it's also very time consuming, mostly if you have 50 different markers. So you'll have a lot of pairs. You can do a lot of 2D scatter plots. It's very difficult to do, very challenging, time consuming, and it's also very sensitive to batch effect. So there you can use another approach. For example, you can use supervised models. In that case, what you do is that you will annotate a few cell types. You will annotate cell types on a few files using manual gating, and then you can train a model and annotate new FCS files. So this approach is faster, at least on the new FCS files. It's reproducible, but it still requires some manual gating on the first FCS files. So it's not really solving the initial issue of manual gating. And again, it's very sensitive to batch effects, mostly if you have a really different type of files in the FCS files that are not annotated. And the third common approach is to use clustering methods. So I think this method is now used quite a lot because it's reproducible and is very good for population discovery. But one major issue is that it doesn't scale very well. Yet in cytometry datasets, we may have hundreds of millions of cells. So sometimes it can be very difficult to run some clustering. And again, it may be sensitive to batch effects. So here on this example, we have a lot of batch effects. And as you can see on the graph on the bottom, we have some clusters. A lot of clusters are really batch specific. So for each cluster, we'll have only one batch inside. So my approach is a little bit different. The idea is to have some expert prior knowledge. So let's say a biologist can say, okay, the plasma B cells are supposed to be CD3 negative, CD38 positive, CD4 negative, etc. So we will fill a big table of size, number of populations times number of proteins. Usually we'll have about 20 to 30 columns. So it's not too long to fill. It's not single-cell RNA-seq data. And we can have a lot of NAs in the table. Of course, we don't know everything. It's not possible to fill the whole table. So we can still have a lot of NA, and the model will be able to work with that. So then, using FCS files and this table, we can train Scyan, which is fast and scalable and can also correct batch effects. Then we'll have our annotation. And for every population, let's say here's a TCD4 cell, we can have some model interpretability to understand why Scyan decided that these cells were TCD4. And we can also run some population discovery to understand why some cells were not annotated by Scyan. So the idea is that if there is a population that is missing from the table, Scyan will say this population is not in the table, and then we can analyze this population, understand to what population it corresponds, and then we can update back the table to be able to annotate it. So in summary, Scyan can do multiple tasks. It's mostly used for population annotation, but you can also use it for debarcoding, batch effect correction, interpretability, and population discovery. So now I'm going to explain how it works. So first, we define what I call a target distribution. And the idea is that we will build this target distribution based on the knowledge table that is filled by biologists. So beside these equations, it's actually quite simple. The idea is that we'll have some population on vertices of a hypercube. Each dimension corresponds to a marker. Here, for example, you have CD3, CD4, CD8, and TCD4 cells are here CD4 positive, CD3 positive, and CD8 negative. So they are here. And each population has a weight that is learned by Scyan, learned by the model. So this is the target distribution. And Scyan, the model itself, is trying to map the actual marker expression of a cell into this target distribution. So the model itself is what we call a normalizing flow. It's a type of deep generative model similar to GANs, let's say. But the difference is that it's invertible, which means that we can go from protein expressions into the target distribution, and we can go then back into marker expressions. And this model will also take as an input some covariates. For example, we can provide some batch information in case we want to correct batch effect. So in terms of loss, this is a typical loss of a normalizing flow. I'm not going to detail it. Just quickly explaining that there is here a determinant of a Jacobian, which is very specific to the normalizing flow, because I mean, we require it to be invertible, so we can compute this Jacobian, and the determinant is actually pretty simple to compute because we have a matrix that is triangular, and so the determinant will just be the product on the diagonal. And so yeah, the loss is very similar to normalizing flows, except that this probability here is a mixture. I mean, the target distribution is a mixture of distribution. So we have this definition for the probability P. And concerning annotation, after training, it's actually quite simple. We take a cell, we project it into the target distribution, and we just assign it to the most likely population. So basically let's say the closest population. So if we have a cell that is here, we'll say, okay, this is a TCD4 cell. Then concerning batch effect correction, what we do is that we take a cell, we project it again on the latent space, and according to training, we don't have batch effects on this latent space anymore because we use the covariates, and the model was trained to remove any impact of the covariates on the latent space. So we don't have batch effect here, and then we use the inverse transformation to recover the original expression, but using the covariates from the reference. So I will say it again. We take the marker expression, we go in the target distribution to remove the batch effect, and then we go back, but using a reference. So that the idea is to project marker expressions into a reference. And concerning the interpretability, the idea is that we can decompose the probability for every population into a sum of impacts of each marker towards the prediction of each population. I'm going to show it in the results. So now concerning the results, this is the annotation benchmark. So here I'm comparing Scyan to 3 different models. One is called ACDC, MP, Phenograph, and they are evaluated on 3 different data sets. One is called POISED, AML, BMMC, on 3 different metrics, F1 score accuracy, balanced accuracy. And as you can see, Scyan, which is in blue, not in cyan, is always above the other methods on all the different data sets. Here, though, the ground truth was manual gating. So people run some manual gating, and we are not, of course, using these labels during training. It's a completely different approach, but at the end, we compare the results to the ground truth from manual gating. Here, this is an example of the annotation of the different models on the POISE dataset. On the left, you have the pseudo ground truth, and on the right, you have the annotation from the 4 different models. So as you can see, the Phenograph model is the one that is the closest to the ground truth, except that you may miss some minor populations. For example, here we missed the dendritic cells and some subpopulation of monocytes, mostly the the intermediate and classical and non-classical monocytes. And here, this is Scyan. So as you can see, it's much closer to the ground truth. And we were also able to annotate more precise population, which are the one here in red, which are more precise than what the author was able to do on using manual gating. We also annotated more cells than the ground truth, because as you can see here on the left, a lot of cells were categorized as unknown in gray, while we have more cells annotated. Then here, this is a computational benchmark. So I compared Scyan to the three same models, Phenograph MP and ACDC, in terms of runtime on the left and RAM usage on the right on different dataset sizes. So we go up to 4 million cells, and Scyan can run in 5 minutes using 4 GB of RAM, while the other models can go up to 7 days using half a terabyte of RAM. Concerning batch effect correction, here we have an example of a dataset with a lot of batch effects. We have 7 different batches. And here is a correction of 4 different methods. SIDAR combat that are not correcting a little bit, but not a lot. So see that is quite weird, let's say. I can't explain why the latent space is so weird, but they have similar latent space in their paper. And harmony, which is very good on this example. But one issue of harmony is that it's not actually correcting the expressions. It only operates on the reduced space. So if we want to go back to a corrected, let's say, corrected CD4 expression, we will not able to have it with harmony. So we will be able, for example, to run some clustering on the latent space, which is very nice. But if we need to have corrected expressions, we'll not have this. And here, this is Scyan. So we have also a good batch effect correction, similar to harmony. But the difference is that we can go back to the corrected expressions because the network is invertible. So it's really possible to recover the corrected expressions. And this graph on the left is summarizing this batch effect correction benchmark. So the ideal model is on the top right. So we want to be really really low in terms of CLEZ, so on the top, and on the right for a better batch effect correction. And as you can see, Harmonia and Scyan are both very good, with Scyan being slightly better for batch effect correction, but it's quite similar. So the difference with Harmony is really that we are able to have some corrected expressions. And thank you, everyone. That's it for Scyan. So don't hesitate to ask questions. I have other slides that I have not shown if you want to go deeper into some details.
That's great. Perfect. Thank you so much for the nice presentation. Questions either on the chat if you don't want to open your mic or anything, or just raise your hand and I can call your name and you can ask questions. But I always have the privilege to ask first. So as long as we don't have anyone, I can go first. So it was great. I have 2 questions. If it's stupid, just let me know very fast, very quickly. Just tell me that's stupid. So the first one is, just to clarify, the markers you are saying, they are expression, like they are markers that they are on the surface, right? The protein markers. That's how we target that's how we recognize the cells, let's say.
Yeah.
But what you're doing is that you're correlating these markers because you know that they're expressed, because that's how we actually do the sorting, right? With expression profile, and then you're predicting it in a later time point.
So can you rephrase it?
Yeah, sorry, because my question is kind of, it goes through the sorting and you know the markers because the biologist actually colors them, right? And then what is the next step?
We don't go to the sorting. here we have the expression for the cells, but we don't know yet for which marker they are positive or negative.
Okay, you don't know them, but the table is coming from the biologists, like which combos they would like to see. I didn't get the table where it's coming from, but you're saying that you have CD4 positive, CD8, but yeah, exactly.
Yeah, it's built by biologists. Indeed, it's which population they expect to see inside all the cells.
Okay, and then you do the prediction based on the expression profile of the single cells.
Yeah, and and we work on all the cells. So typically all the immune cells or immune plus tumor cells, let's say. So we don't have sorted cells.
I see. And the validation then, how does it come? You know the labels, or how do you know then that you predicted kind of correctly? What's the benchmark that you predicted correctly the surface markers?
I mean, we are not predicting. You mean predicting the cell types?
No, you mean the cell type. Sorry, that I get confused with the table. Yeah, that you predict the actual cell types. I guess with the labels, right? You have some labels. Yeah.
Yeah. I mean, we have this benchmark of annotation, but in practice, when you don't have ground truth, we just look at, let's say we can have a look to heat maps of expressions per population, and a biologist will be able to say very quickly if it looks good or not.
Yeah, true. And actually, this one should have been Scyan. I mean, come on. So you should have colored it like that. Okay, and I have another one. It's awesome. So once you, no, because you said that it's not correlate. Maybe it's already canceled. So no, my question would be if you can actually come up with maybe better combinations of markers, but you don't really associate them in the end of the day, right? Do you? Like we have a combination of markers expecting to see a specific cell population, but you predict that with the expression. So I was thinking if you can come back and say, this is a better expression, this is a better combination of markers to predict this population.
Yeah, of course. Since Scyan is very can learn very quickly, it's definitely possible to have many iterations. We run Scyan, and okay, the table is not perfect. Maybe we can update it. So you go deeper into a specific cluster that is not nicely annotated, and or you ask some people, do you think I'm right on this part of the table? And then you update it and you run Scyan again. So it's very usual to run it multiple times.
Okay, that's really cool, I think. Maren, thank you for the answer. Maren, go ahead.
Yeah, thank you, Quentin, for the very nice talk. I was wondering, you're saying your table, like your expert knowledge is essentially binary. So you say like it's not available, it's present or absent, or like positive or negative. How do you deal with like continuous values? So for instance, when we take the monocytes example that you mentioned, so the distinction for classical to intermediate to non-classical monocytes, essentially a continuum from of the marker of CD14 and CD16, if I recall that correctly. So how well can you actually make the cut, and how is this represented on your latent space? Because you say like your latent space, essentially like all populations should be on the edges of your hypercube, but if you have like a continuum, as in the case of monocytes, they should be like organized somewhere on the edges, I assume. Do I take that correctly?
It's a very good question. So indeed here I put the population on the vertices to make it easier, but we may have some values that are not -1, 1, or NA. We can have, just on this example, we can have what I call low values or medium values. We can put, for example, 0, or I mean anything that is between -1 and 1. And the result is that we'll have one population that will be on the edge of the hypocube instead of being on a vertex. So in that case, we can have a population that is here in the middle. So this is one thing that we can do. And the other thing, if we have a marker that is really like really a continuum, then you can change the standard deviation for this marker. This is the standard deviation that we use here in the target distribution. So when it's very clear cut, when you really have two different peaks in the distribution, we'll have this standard deviation to be very low. But for continuum of expressions, you can choose it to be very high. So in practice, to make it simple to train, we don't want to choose this standard deviation for each marker. So we just say if we have a continuum, if one marker is a continuum of expression, we have an argument, continuum markers, it's a list of marker names, and we just provide it inside this list, and it will update the standard deviation for this specific marker.
Okay, makes sense. Thank you.
So two solutions, you either provide this marker in the continuum marker argument, or you can change the table to have a 0 value or 0.5 something.
Okay, thank you.
Severin?
Hey, thank you for the nice talk. This might also be a very naive question, but from what I hear from a lot of people is that when you get like this kind of data from other labs, they usually have sometimes a different set of markers that they observe. Does your model in the batch correction also account for this kind of data sets that you can integrate those?
So indeed, if you have a different panel of proteins measured, you will have to train a new model with a new table, which is indeed a limitation of the model. Yes, if you have like dozens of data sets with different marker measured, you will need to have one model per data set.
Okay, thank you.
So what are the next steps for Scyan? It's published, right? I'm sorry, I did not recall.
Yeah, it's published. And the next steps may be to test it more on some spatial proteomics data. So for now, it's mostly tested on flow cytometry, spectral flow, and mass cytometry, but not a lot on spatial. And the expression are a little bit different. I mean, it's less clear-cut than mass cytometry. Here on this scatter plot, we can definitely see that we have this CD4 CD8 negative population, we have this CD4 CD8 positive. We really see a difference on so on imaging mass cytometry data. We don't have such clear cut here. So the distributions are different, and Scyan may need to be adapted. So I think that mostly this prior standard deviation must change. So I think it can be, if we set it to 0.5 or 1, so it's a hyperparameter. So if we decide, if we increase it, maybe it can deal with spatial proteomics, but it has to be tested.
No, that would be very cool actually, if you make it work. Okay. Let's see, are there any more questions? Raise hands or put it on chat. Anything you would like to share that you didn't share? Because maybe I told you to do it in a short period of time, maybe you have more things to say. You're more than welcome.
Yeah, sorry, I was very quick, so
No, it was
I
fine.
think
No
it was
worries.
less than 15 minutes, so maybe I can show this slide. Okay, so this is about the model interpretability. So here on this example, I removed some populations from the table, especially I removed intermediate and non-classical monocytes. So Scyan was not able to predict them, and this is what we can see here on the left. We can see that we have some gray cells, some unknown cells, and then we can analyze these gray cells and try to understand which population they may be. So for that, one interesting graph to plot is to look at all the probabilities of assignments to each population. So for example, for this gray cell, Scyan is saying that the classical monocytes are the most probable, then the classical monocyte CD14 mid, then the dendritic cells, etc. So we have a spectrum of probability for each population. And for each specific population, let's say classical monocytes, we can decompose it into probabilities for each marker. So basically, when it's yellow, it means that the expression is in accordance, is consistent with the table. And when the expression is purple or dark, this is not consistent. So here you see that the gray population is consistent with the classical monocytes, except for CD14. And you can see here that the CD14 expression is negative. So you can understand that the population may be related to intermediate or non-classical monocytes. So then you can update the table and you will be able to annotate these populations. And you can do that for many different populations to go deeper into the annotation and annotate here, for example, TCD4 EM, CD27 pose neg, some NKT CD4, CD8, etc. So this is how we go deeper into the annotation.
That's actually pretty cool, and it kind of answers exactly what I was looking before. I didn't phrase probably the question very well, but also it's very interesting that you're saying that it aligns with the classical monocytes, but the CD14, right? So you we could say that you are able to even capture subtypes in a way, like cell states, I guess, not subtypes, maybe, right?
Yeah.
Very cool.
And maybe a last slide, since I still have some time. So this is concerning debarcoding. So I don't know if you know what this is, but I'm going to be very quick. So the idea is to remove batch effect. And for that, you can do one big experiment with every cells mixed into one big experiment. But then you will not know which cell comes from which patient. So for that, we create a barcode for each patient. For example, the patient A2 will be 110100, which means that for each cell of patient A2, we will add or not some specific reagent. And then after the experiment, if we see that this specific cell is positive for the first reagent, the second, not the third, the fourth, etc., then we can say this cell belongs to the patient A2. But then so this process is called debarcoding, so retrieving the patient ID from the barcode. And Scyan can also be applied to this. It's a very straightforward case because we already have the knowledge table. So in that case, the knowledge table will be the barcode. And we can retrieve, as shown on this example, the patients from the cells. So that's it.
Good. So any more questions? I don't see anything, and I have shared all your details. You're also on Zulip. I've shared LinkedIn, Twitter, GitHub, everything. So feel free to reach Quentin and ask more questions, and especially on Zulip. And if you want to continue also the conversation, you can also continue on the channel that I have actually put the talk today, and I have shared your GitHub and everything. Thank you so much. The presentation was great, and thanks everyone for joining. And see you in 2 weeks.
Thank you so much. Bye.
Bye.